Hello, everyone. I'm Dr. Edward Hoffer, the speaker for today's webinar. I will be presenting material for about 40 minutes, and we will then have a 10-minute Q&A session. Please jot down any questions or comments that you have. We've got quite a bit of material to cover, so I'm going to try to go through it fairly quickly. So let's get started with today's topic. Hyperlipidemia, statins, and beyond. And away we go. It is visible now?
Good. Yes, doctor, you can start the webinar.
All right, thank you. The obligatory commercial disclosures, I have none. I have never taken a penny from Big Pharma and have no plans to do so. The whole issue of cholesterol and coronary disease is actually remarkably new. The connection was only established from late in the 50s through the mid 60s. A seminal paper from the Framingham Heart Study in about 1964 established that indeed high cholesterol was a major risk factor for coronary events. Unfortunately, in that period of time, attempts to reduce heart attack by lowering cholesterol were not terribly successful. Major reason being that the medicines available at that point were poorly tolerated and not terribly effective. I'm not sure if many in the audience still remember niacin or cholestyramine, but many of my patients, after an initial attempt, would say, "Thank you, doc, but I'd rather risk a heart attack than take this for the rest of my life." The major reason why diet alone is almost never successful is that the cholesterol manufactured in the liver is the major determinant of circulating cholesterol, much more important than dietary cholesterol. I'm not saying that low cholesterol diets aren't to be advised, just warning that they are rarely terribly effective. Liver cholesterol synthesis, in turn, is mediated by a multi-step process that we'll show a little later, but the enzyme HMG-CoA reductase is a key step in the manufacture of cholesterol in the liver. And so logically blocking HMG-CoA reductase will suppress liver cholesterol synthesis and in turn lower cholesterol. The initial work on HMG-CoA reductase inhibitors was begun by Sanyo, a major pharmaceutical firm in Japan, in the 1970s, but they never pursued it terribly actively because of animal toxicity that showed up early. In this country, some investigators working in the Merck Research Lab discovered lovastatin, somewhat analogous to Fleming's discovery of penicillin. They found it in a fermentation broth of aspergillus. They did human trials in healthy volunteers beginning about 1980, but after Sanyo's product Compactin was withdrawn due to toxicity in animals, Merck suspended trials. On a compassionate use basis, small-scale trials of lovastatin were begun about 1982 in patients who had severe hyperlipidemia, familial hyperlipidemia. And these very small trials showed that the product worked and that in fact in humans it had almost no side effects. Clinical trials expanded and showed the same thing: excellent tolerability, excellent cholesterol lowering effects. And the first statin was approved for clinical use in this country in 1987. At the top recommended dose of 80 milligrams a day, lovastatin reduced LDL cholesterol by 40%, dramatically better than any of the old, less effective and less well-tolerated drugs. And lovastatin became a smash commercial hit for Merck, reaching annual sales of over $1 billion. And of course, that opened the floodgates to a number of Me Too drugs. And there are now at least 7 available for sale in this country, and possibly 8 if you look worldwide. Okay, lowering cholesterol is great, but as I have said repeatedly, cholesterol is not arsenic. You don't die of cholesterol, you die of coronary disease, which in turn is mediated by cholesterol. So you've got to show that not only does the drug lower cholesterol, but that it prevents coronary disease and prevents deaths. And the landmark trial in this was the 4S study done in Scandinavia. They took over 4,000 patients who were by definition high risk. They had known coronary disease. These were patients who'd suffered a myocardial infarct or had ongoing angina. All of them were given the standard low-fat diet of the day, randomized to either simvastatin or placebo, and followed up for 5.5 years. In the doses used, simvastatin reduced LDL cholesterol by 35% compared with the placebo group, and there was a quite statistically and clinically significant drop in the death rate. It was reduced from 12% in the placebo group to 8% in the simvastatin group for a relative risk ratio of 0.7. The decrease in coronary deaths was even more dramatic, and there was a reduction in the need for coronary bypass surgery. So a clear and absolute benefit in this group. Dropping the bar a little bit, we look at the West of Scotland trial that was reported out about a year later. They looked at 6,500 men who had hyperlipidemia with a mean cholesterol of 272, but no known coronary disease, a middle-aged at entry, and followed for almost 5 years on either pravastatin or placebo. And again, coronary events, non-fatal MI were reduced 31%. All deaths were reduced by 22%. And the absolute reduction, as you can see, was not as dramatic as in the 4S trial, but almost 1% less. Dropping the bar even lower, the AFCAP study enlisted active duty Air Force members, and this was the first trial to include women, though again, the majority were men. These were healthy adults with what was felt to be at the time a normal cholesterol for the US in the 1990s. Mean was 221, and they were treated with lovastatin. It was started at 20, could be increased to 40, and this again was compared with placebo, follow up about 5 years. And the outcome, which has become fairly standard, the abbreviation is MACE, Major Adverse Coronary Events. And they included fatal or non-fatal MI, hospitalization for unstable angina, or a sudden cardiac death. And the primary endpoint was reduced in a similar manner, 37%. Obviously, this was a lower risk group, and the absolute lowering was smaller. And I would like to call your attention to the difference between absolute and relative benefits. Pharmaceutical companies love to talk about the relative benefits of their product compared to others, because these are always more impressive numbers. You know, if you say that something is reduced 35, 40%, boy, that's really impressive. But I caution you, especially if you're dealing with lower risk patients, to think in terms of absolute benefits. So let's compare the 3 trials in 3 different risk groups. 4S trial, known coronary disease, clearly very high risk group. And the events were reduced from 28% to 19.4%. So an absolute dropping of 8%, 8 per 100 people treated. West of Scotland trial, a moderate risk group, and again, similar relative risk, but the absolute risk reduction was a little over 2%. Similarly, AFCAP's trial, a low risk group. And again, a lowering of similar percentage wise, but the absolute difference was smaller. Even more contrast when you look at deaths. And when looking at any study of any product, the first number I like to look at is deaths. Most everything else is potentially problematic. If you read the fine print, you'll see that in many cases, a committee will adjudicate whether somebody had a heart attack. And at least to my way of thinking, you don't need a committee to decide if somebody's alive or dead. So start with deaths. Those are hard, about as hard a number as you can get. High risk group, 12 to 8%. So for every 100 people you treat, you are keeping 4 additional people alive. Quite important to most patients. When you get to a less high risk group, you're getting about 1% reduction in deaths. And when you get down to the low risk groups, you're getting 0.4 absolute reduction. So very similar relative reduction, but much lower absolute reduction. A recent meta-analysis looked at all the statin trials, and they came up with not a surprising conclusion, which is major cardiovascular events were reduced similarly at all risk levels. And in fact, the lower risk group, you might have had a larger relative risk reduction. But when you're dealing with a patient in front of you and you're dealing with somebody who is borderline as the indication, remember to think absolute. You know, how much are you actually benefiting this patient by prescribing a drug? All right, statins are, to my mind, one of the most successful group of drugs ever devised, perhaps second to antibiotics, but they do have the downside, and the downside is side effects. And this, if not a concern to you, I can assure you is a concern to your patients. And if you go on social media, you'll discover enormous amounts of discussion about real and putative side effects of the statins. First concern to come to public attention was cognitive impairment, dementia, and in fact, in 2012, the FDA put a black box warning on all of the statins, and at the time, that did lead to decreased prescribing. The following year, an examination of their AIRS, the Adverse Event Reporting System, claimed that this should not apply to hydrophilic statins like pravastatin, but only to the lipophilic statins. The best study that I'm aware of that looked at this was a very large review and meta-analysis that was printed 2021. And they looked at 24 studies with almost 1.5 million enrollees. And they found that there was absolutely no association of statin use and cognitive impairment. And I think that this is something that you can count on as being established, and reassure your patients that this does not seem to be a real phenomenon. The reason that Sanyo never marketed Compactin was because of fairly significant liver toxicity in some of the early animal studies. And because of that, the FDA mandated initially that all the statins have a package insert recommending that liver function tests be monitored periodically. This proved to be so rare that in fact, this was removed from the package insert in 2012. It is real, but it's infrequent. Somewhere on the order of 1% of statin users will have modest elevations of liver enzymes. And if you continue the statin, about 70% of these tests will return to normal within 2 to 3 months. And they do not require anything more than monitoring. Serious liver damage does occur. I've been fortunate never to have had it occur in my personal practice, but I'm aware of at least one patient who did have very severe liver damage. It is estimated to occur in roughly 19 per 100,000 users and can have either a hepatocellular or an obstructive pattern and this does require permanent cessation and not using any other statins. Acute liver failure has been estimated to occur in about 1 in a million. Take home from this is don't worry about the liver, don't check routinely, but if anybody has any symptoms at all, that's the time to check their liver function. Diabetes is a more recent concern, and diabetes is real, and there's a pharmacologic, physiologic explanation. Statins do decrease insulin secretion by beta cells. They increase peripheral insulin resistance, and there is a larger relative but small absolute increase in the new diagnosis of diabetes in patients put on statin. This was looked at specifically in the Jupiter trial of rosuvastatin. There was a 24% relative increase in new diagnoses of diabetes. The absolute increase was a more modest 0.6% of users. Why do people with diabetes die? The vast majority of them die from cardiovascular conditions, and most studies have shown that, in fact, the benefit from lipid lowering vastly exceeded any concern about increased diabetes. So again, I think something that needs to be kept in mind, but should not dissuade you from prescribing the drugs. The elephant in the room, muscle. Rhabdomyolysis does occur. It's exceedingly rare, less than 1 in 100,000 per year, but is obviously very serious, can lead to acute tubular necrosis. Still very uncommon. There is an immune-mediated necrotizing myopathy that occurs in estimated 2 to 3 per 100,000 users, and you'll get CK elevations, you'll get abnormal EMGs, and this is a cross immunity between the statins and muscle. There is a direct toxic myopathy from statins with where the CK is markedly elevated, and you get proximal muscle weakness. This is so rare that there are no good estimates of the actual incidence. There's been isolated case reports. So these things are real, they are rare. What is not rare is myalgia. And some 9 to 10% of patients who are begun on a statin do complain of muscle aches. This has been looked at 6 ways to Sunday, and it is in the majority of cases a nocebo effect. Crossover trials have shown that patients had a similar degree of complaints of myalgia, whether they were taking a statin or a placebo. I can testify to this personally. When I finally went on a statin, I began to get achy, was sure it was a statin until I realized that I had also decided to up my weights at the gym by 25%. And in fact, that was why my muscles were achy. When I backed down on the weights and continued my statin, the muscle aches went away. It is a problem that clinicians have to deal with, and is probably the #1 reason why your patients may stop taking their statin. How do you handle it? If the patient complains of muscle aches, first thing you do is measure the CK. And if that is normal, you can reassure the patient that this is nothing serious. And if they still want you to do something, step 1, lower the dose. If you've started them on 10 of atorvastatin, cut it to 5. If they still complain of muscle aching, I would switch to a different statin, and again, using pharmacologic thinking, try switching them from one of the more commonly used statins to pravastatin, which is water soluble, and you can honestly tell them this is less likely to do it. If they're still complaining of muscle aches, add over-the-counter CoQ10, which has some very soft data behind it saying that it's helpful. And if it persists, then you simply have to give up and use an alternative drug. Again, if your patient says they hurt, and you can't talk them out of that. How large is statin? There are two approaches to alternative protocols, either treat to target or use a maximal available dose. One study looked at 2 different doses of atorvastatin, and they felt that, an arbitrary dose of 80 milligrams of atorvastatin was clinically more beneficial. That was less clear given the comparator, which was a lower dose simvastatin. Study that was just reported out a little over a month ago looked at 4,400 patients with coronary disease, and they were given either a high dose of statin arbitrary or had the statin titrated to get their LDL below 70. And there was no significant difference. There was a trend towards fewer events in the titrated group, but the bottom line is it was not inferior to using an arbitrary large dose. And the pharmacology of most statins is unusual in that the dose response curve is pretty flat, that if you're going to get the most bang for your buck out of your starting dose, and every time you double the dose, you get less and less return in terms of lower cholesterols. So start on the low side, measure, and go up if you need to. Some of you may be asking yourselves at this point, why is this guy talking about statins? I know everything there is to know about statins. I've used them forever. And the reason I am starting off with statins, and we will end up finishing with them, is the data that I'm showing you right now. Data from the National Health Study looks at a representative sample of people all across America, studies a whole variety of things. And they looked at 3,400 middle-aged people who had LDLs over 190. And I think everybody in this country, everybody on this webinar would agree that an LDL over 190 is really bad and should be treated. Okay, but only a quarter of these patients were on any drugs to lower their cholesterol. Not surprisingly, Blacks and Hispanics were even less well treated than whites, but even among Caucasian patients, a mere 28% were prescribed statins with LDLs that were dramatically elevated. If you look at at their risk group, factor in things like smoking and family history and blood pressure, they were stratified into 3 different risk groups. And even among the highest risk group, only 35% of these people were being treated. So, you may think that this is an answered question, but I can assure you, if you look across the country, that these drugs are vastly underused at this point. The drug that kind of fell by the wayside is ezetimibe or Zetia, which inhibits intestinal cholesterol absorption. Since, as I mentioned in the beginning, liver synthesized cholesterol is more important than dietary cholesterol. Not surprisingly, it is not as effective as the statins. You get about a 17% reduction in LDL cholesterol, and it's used at a fixed dose, 10 milligrams a day. Using more doesn't get you any further LDL lowering. Until the last few years, Zetia was probably the only option for the statin intolerant, but it's now primarily used as an add-on. And that's because the clinical effect is pretty modest. A IMPROVE-IT trial compared Simva 40 with Simva 40 plus Zetia 10. and the hazard ratio did reach statistical significance, P of less than 0.02, but the hazard ratio was only modestly reduced, and it took almost 5 years for the curves to separate out to a significant level. So it is, it is a drug that has effect, but has a modest effect. The newest trial looking at the value was a trial done in South Korea, which looked at some 3,800 patients with coronary disease and compared rosuvastatin 20 with rosuvastatin 10 plus CD10. And this would fit in with your patients who are somewhat statin intolerant. You can drop the dose of the statin, add on 10 milligrams of Zetia. And there was in the combination group, there was a non-significant trend towards fewer adverse events in the combination group. So I think we can safely say that a lower dose of rosuvastatin plus ezetimibe is non-inferior to a higher dose of the statin. All right. The new stuff, the sexy stuff we're going to get into. The newest drugs that are became commercially available over the last couple of years are the PCSK9 inhibitors. The reason we use the abbreviation is because the actual name is quite a mouthful: proprotein convertase subtilisin kexin type 9. It's encoded by a specific gene on the human chromosome 1, and it reduces LDL receptors on the liver cell surface, which induces increased liver synthesis of LDL. So increased PCSK9 leads to elevated LDL cholesterol. There's a small group of patients with familial hypercholesterolemia who have it because of a gain of function of PCSK9. This is what, in fact, led to the study of blocking drugs. And inhibiting PCSK9 leads to lower LDL synthesis in the liver. There are 3 available products that target PCSK9. 2 are very, very similar to each other. The Repatha and Praluent are the trade names for 2 monoclonal antibodies. They are both given subcutaneously, either every 2 weeks or every 4 weeks. Very similar in action, very similar in results. Inclisiran is a different approach, different molecule. It's a small interfering RNA, which inhibits intracellular synthesis of PCSK9. And it's given at baseline, given again in 3 months, and then given every 6 months indefinitely. And we'll talk about these a little bit more. This is one of the antibody drugs showing you what it is doing, that it binds PCSK9, affects the LDL receptors on the liver, and reduces the synthesis of LDL cholesterol. Dramatically effective in terms of lowering LDL cholesterol. All 3 available products, whether used as monotherapy or added on to a statin, will reduce your LDL by about 50%. The dosing, pretty standard with alirocumab. You start with 75 every 2 weeks. If you don't get the results you want, it can be increased to a maximum of 150 every 2 weeks. And if that is working, you can switch to 300 every 4 weeks. The Velocumab, again, starts off Q2 weeks. You can also use a slightly larger total dose on a monthly basis. They work equally well. Inclisiran has a single dose, comes in a single injectable vial. Give it once, give it again in 3 months, and then every 6 months. They lower cholesterol, unequivocally proven. How well do they work in terms of helping your patients? There have been a couple of large trials recently reported. The Odyssey trial looked at some 19,000 patients who had 40 or older who were hospitalized with an acute coronary syndrome, either unstable angina or MI within the prior year, and who had an LDL of 70 or greater despite a maximum tolerated dose of statin. And this is going to be a repeat theme in these drugs, is all of them to get into the trials had to be on a maximum tolerated dose of statin. LDL was reduced from 93 in the placebo group to 40 in the antibody group, as would have been predicted. Follow-up mean a little under 3 years, and major adverse coronary events were reduced 15%, from 11.1 to 9.5, so an absolute drop of about 1.6%. Number of deaths, where there was a trend to fewer deaths, but it did not reach statistical significance. Remarkably well tolerated, as you'd expect, with some injection site reactions. There was some increase in nasopharyngitis. There's no evidence of cognitive impairment, liver impairment, or myalgia, and there were no drug-drug interactions found. So very well tolerated, very effective at lowering cholesterol, moderately effective at reducing coronary events. The Fourier trial, even larger, so over 27,000 patients between 40 and 85 who had clinical coronary disease, LDLs 70 or greater on a maximum tolerated statin, and they were followed up not quite as long, for a mean follow-up of a little over 2 years. Again, you saw the dramatic drop in LDL cholesterol, 92 in the placebo group down to 30. Major adverse cardiovascular events reduced by a similar degree, 15%, and an absolute 1.5%. And deaths, so there was no significant difference. There was some number of secondary analyses done of this large trial. They looked at people who had metabolic syndrome. They had a similar LDL lowering, and they had a higher baseline event rate, but a similar lowering. There were the Fourier open label extension, followed some 6,000 patients from the original Fourier trial, about half of whom had been on active drug, half on placebo, followed them for another 5 years. So they had the groups that had been on active drug the longest that were followed for almost 9 years. After 12 weeks on active drug, there was a median LDL of 30. 2/3 of the patients had an LDL below 40. Same story, 15% reduction in major adverse events, some 20% lower risk of MI, stroke, or cardiovascular death. So these drugs work, and they seem to keep working. One issue that was brought up when people looked at the sort of results you could get from the antibody drugs was, is it possible that you're taking your cholesterol too low? Cholesterol is not arsenic. Cholesterol is an important compound that's used in the human body. It's part of the cell structure. Cholesterol is a building block for a number of sex hormones. And so there was at least theoretical concern that dropping the LDL cholesterol as low as could be done might in fact be dangerous. Looked at the longer term, the open label extension of the Fourier trial, 24% of these patients had LDLs below 20, and fully 2/3 were 40 or below. So, these were dramatic lowerings of cholesterol. And when they looked at the different groups by the level of cholesterol lowering, the lower the LDL cholesterol, the fewer primary, secondary endpoints, and there was no increased risk of side effects. So it does not appear, in fact, that there is any harm from getting LDL cholesterol down to these basement levels. Inclisiran also targets PCSK9, but does it through a different mechanism. One of the attractive features of the drug in the initial working was that it is very specific to liver cells. If you give an IV infusion and look 24 hours later, you can find none of the medication in the serum. It's all gone intracellularly, and it decreases both intra and extracellular PCSK9. And because of its mode of action through RNA, it gives a very long-acting lowering of LDL cholesterol. And its maintenance, it's used every 6 months. And there's pretty good evidence that if you're using it for primary prevention, you can in fact probably give it once a year. So for people who don't like the idea of getting an injection every 2 weeks or every 4 weeks, this is a pretty good option. Much smaller trial, the ORION trial, but again, it looked at 2 groups of patients. It looked at those that had stable coronary disease or who had LDLs greater than 70, or high-risk patients who had LDLs over 100, again, on a maximum tolerated dose of statin. Their follow-up was shorter, only a year and a half. LDL cholesterol reduced by 50%, and adverse events, cardiac adverse events, reduced by 26% relatively, and a fairly impressive 2.3% absolutely. And it was dominated by a decrease in cardiovascular deaths, which is my favorite hard endpoint. Well tolerated, no liver, kidney, muscle toxicity. And when the open label extension showed that the cholesterol reduction was long acting, that after 4 years after the initial trial, LDL was still reduced 44% from baseline. So we now have 3 good drugs. This is a little schematic of inclisiran's working. I don't think it adds much. The newest kid on the block, the newest drug that's been approved and shown to be clinically useful, is bempedoic acid, brand name Lexletol. And this is a prodrug which is inactive until it gets into the liver, where it's converted to its active form, and it inhibits an earlier step in liver synthesis. If you, this is a little schematic of the production of cholesterol in the liver. You can see the gray arrow where the statins work, and the green arrow is where bempedoic acid works, an earlier step in the cholesterol synthesis sequence. It's taken orally, which obviously is more appealing to many people than injections. It is not nearly as effective at lowering LDL cholesterol as the 3 injectable drugs. Compared with placebo, it decreases LDL cholesterol about 21%. For those who are fans of the pleiotropic action of statins, it's an interesting side note that bempedoic acid decreases high sensitivity CRP by 26% compared with placebo. There is a fixed dose combination of bempedoic acid with ezetimibe, and that lowers the LDL cholesterol a more impressive 38%. But at the moment, there are no clinical trials of this combination. The CLEAR trial was reported at the last ACC meetings and published a little over a month ago in the New England Journal. A large trial, 14,000 patients who had either known coronary disease or considered very high risk, and who were statin intolerant, who refused to take statins of any sort, or either because of objective or subjective reasons. Mean entry level LDL cholesterol was 139. Similar study endpoints, they followed up for 3.5 years. And the LDL cholesterol, as predicted, was reduced by a mean of 21%. And the primary endpoint was reduced 13%, with an absolute reduction of 1.6%. very statistically significant. There was no difference in deaths, and the major side effects reported were small increases in symptomatic gout and symptomatic gallstones. So is this in the same league with the injectable drugs? No, it is not, but it is an option for those who will not take a statin and at this point will not take an injectable drug. Wonderful drugs, lower cholesterol, reduce heart attacks, reduce deaths. Is there a problem? Yes, and the problem is their side effects on the wallet. As you're, I'm sure, well aware, generic statins are now available that are so cheap that most insurers don't even put on a copay. If your patients do have to pay for them, they can probably get them for about $5 a month. The two antibody drugs are both in the $6,500 a year range, as is inclisiran, bempedoic acid, $4,500 a year. So these are not insignificant costs. And it's particularly a concern when you figure that your average coronary patient is frequently taking multiple drugs for other conditions and really is hurting if they don't get this covered by their insurance. So a side effect for the prescribing doctor is you've got to go through hoops to get their insurance to cover it. And it would be nice if it were easier, but it isn't. Where do the experts say we should use them? These are the published guidelines from several years ago. They will probably be updated, but haven't been. And they basically say that use of these drugs should be considered in people who are at high risk, who are on a maximum tolerated statin plus ezetimibe. And you can look at different groupings, but the idea is the same, that people who are not at goal, who are either known to have coronary disease or at high risk of coronary disease, and are either on a statin but not at goal, or simply will not take a statin. What is very encouraging for your injection intolerant patients is there are a number of oral PCSK9 inhibitors that are being studied. There are at least 3 actively being studied that I'm aware of by Merck, Pfizer, and Novo Nordisk. The Merck drug is furthest along and then had a dose finding phase 2 trial reported at the ACC meetings, and it was published in April, and they were patients who, again, had either clinical coronary disease or were very high risk and had LDLs over 70. And they were given 4 different doses of the oral drug, and it worked at all doses. It looked as if there was a plateau somewhere around the 12 milligram per day dose, but wonderful reduction in cholesterol. At this point, no clinical data at all. The drug will be starting a large clinical trial, and I don't expect reports will be out for 2 to 3 years, but again, well tolerated, very effective at lowering cholesterol. All right, to sum up, step 1, you've got a patient in front of you, think globally, think risk. If you have an active patient who doesn't smoke, who has a normal blood pressure, has no unusual family history, and a modestly elevated cholesterol, you need to have an informed discussion of the fact that this may indeed lower their risk of heart attacks if you put them on a drug, but that their absolute benefit is going to be modest. And you'll have patients who say, yeah, I want to have my heart attack risk reduced by 30%, even if it is only from 0.6 to 0.4 and go right ahead. You've got good drugs available. If they say, I hate taking medicines and, how much is this really going to help me? You can assure them that it's not unreasonable to just follow along, do their best with diet and keep an eye on it. If you're going to treat, you should always start with a low dose statin. I don't think it matters a whole lot which one you pick. My favorites tend to be the rosuvastatin or atorvastatin, certain psychological effect that you're giving people a low number of milligrams, and so they feel more comfortable with that. But whichever drug you start with, start low. Remember the fact that starting somebody on the lowest effective dose is going to give you 50–60% of the benefit that you'll get from the maximum allowable dose. So avoid complaints, avoid muscle aches, start with a low dose and measure and go up as needed. If people feel well, there's no need to do routine testing of muscle or liver, but if they have any sort of side effects, however non-specific, I would check CK and liver enzymes. If you have significant elevations of liver enzyme or CK, then the statins are not for them. Stop the statin, use an alternative drug. If their problem is primarily muscle aches, their CK is either normal or minimally elevated, I would switch to a different statin or a lower dose. If you've gotten up to 3 or more statins and patients are still not happy, you can switch them to oral bempedoic acid. And if there's still not a goal with that, that's the time to go to a PCSK9 inhibitor. If they are on a statin and they don't reach your mutually agreed upon goal, which would be below 100 as primary prevention, below 70 for secondary prevention, first step is add ezetimibe 10 milligrams, inexpensive, well tolerated. If that doesn't get you to goal, then it's time to either add on or switch to a PCSK9 inhibitor. I have done a lot of talking, and I will now open the floor up for questions. The webinar was sponsored by MDforLives, a premier healthcare market research firm. Doctors can register, become part of the community, take surveys, participate in quizzes. So the sponsor urges you to sign up. If you would care to ask a question or make a comment, please raise your hand and we'll try to get to all of them. Shoot.
Dr. Hoffer?
Yes.
Thank you so much for an excellent talk. Alison Muffler here from Kingston, Ontario. I like the approach of trying to start, with the lowest effective dose and then escalating. How do you handle patients who have symptoms despite stopping the medication, but they still, strongly believe that statins are causing their myalgias? Because there's a lot of media and press about the statins, unfortunately, that is making it difficult for us to start on patients. How do you handle that in terms of discussion and counseling?
Well, I try to, and now there is really good data saying that a lot of this is nocebo effect. And I tell people my own little anecdote about my own myalgia that I attributed to the statin. And then I go over with them the fact that people have done studies showing that whether the patients were taking a statin or taking the sugar pill, there was an equal amount of muscle aches, and asked them if they're willing to try a lower dose. And if they say, no, I'm convinced this drug did it to me, then I can, with science behind it, say, well, you've been on a fat soluble drug. We're going to switch you to pravastatin, which is water soluble and is less likely to do this. And I would say 2/3 of the time, after 2/3 of the time, they're quite comfortable on pravastatin, having gotten muscle aches on one of the other statins. If they insist, and we've tried that, and we've tried CoQ10, and they still say, this stuff is awful, I ache, then they ache and they're not going to take it, no matter, and there's no point in you saying it's all in your head, because they're going to say that doctor doesn't understand, and they stop the drug. So I would say at that point, that's the time to use one of the alternatives, and hope you can get their insurance to pay for it. Say 4 years ago, 5 years ago, all I had to offer them was Zetia, which was only minimally effective. Now we do have more effective drugs. So, if you've gone the extra mile, sat down with them, tried to work with them, and they insist this drug is awful and I hurt, and all my friends tell me that it hurts, and I read it on Facebook, well, then no matter what you say, they're going to stop. So you may as well work with them and try to find one of the newer agents that they can afford. One thing I didn't touch on because of interests of time, but we have a few minutes, is Lp(a), which has now been recognized, I think, convincingly as a risk factor for coronary disease. One reason I didn't mention it is we have no available therapies to lower it. But it is something, if you've got a patient who's got a nasty sounding family history and their basic lipids don't look too bad, I would definitely measure an Lp(a) and give them the one thing we do have available. Start treating their LDL cholesterol. One thing about Lp(a) is it seems to be genetically determined, so you don't need to keep measuring it. Measure it once, and that'll give you all you need to know. So again, if you're on the fence with somebody as to whether they should be on lipid lowering drugs, that's an extra piece of data that you can throw into the mix. Thank you all for being part of the webinar. We hope to see you again. If you want to reach out to me directly, you can email me. It's ehoffer@gmail.com. And thank you for participating.